Hello everyone, my name is Rafi Karim and I am a fourth year biomedical engineering student at RIT. Today I'm going to be presenting at the RIT Undergraduate Research Symposium 2021. The title of my research is Lysis-free and multiplex detection of E.Coli using the spin chip. And the name of my advisor is Dr.Ke from the Mechanical Engineering Department. And for the last few months, I've been conducting my research under him at his lab called the 3N Lab. And you can read about more about our research at www.3n@rit.com. So for the abstract of this project is that it's basically a subsection of Dr. Ke's NanoBiosensing research and the lab on a chip concept for the detection of bacteria with just a use of around slide spin chip rather than using a huge expensive devices. For this particular project, we are detecting the B-Galactosidase, which shows Lac-Z gene activity in E. coli with the use of Fluorescein Di-β-D-Galactopyranoside. So this is basically the prototype of the spin chip. It consists of two circular round bottom microtiter plates that are facing each other. And on each side, it has 60 wells or reservoirs. And there's also a thin layer of Teflon coating between them to basically prevent leakage. And this chip was designed by my good friend Jimmy He, who's a mechanical engineering student and also works at Dr. Ke's 3N Lab. So this is the CAD model, including the clamp. And then it's basically, for up next is an experiment that we ran using simple dyes, red and green, to kind of like demonstrate the functionality of the mixing process. Up next, I'll show you a short video to demonstrate the functionality of the spin chip. We'll pause my video.
Hey everyone, my name is Rafi Karim and we are here at the 3N Lab, and today I'm going to show you or demonstrate you how the spin chip actually works to detect E. coli. So first of all, we're going to sanitize our hand with some IPA, and now I'm going to show you what the spin chip actually is. So it basically consists of two 96 or in this case 60 well plates on each side, and it's coated with Teflon to prevent liquid adhesion basically, or leakage in this case, and improve the hydrophobicity. So now what I'm going to do, or what I did, was dilute some FDG, which is our fluorescent substrate reagent, in some PBS. And what FDG stands for is Fluorescein Di-β-D-Galactopyranoside, and that's the substrate reagent we're going to use to detect E. coli. So initially what I'm going to do is take 1,000 microliter pipette, and did I already protect it? Just to make sure, and take 200 microliter sample, or 200 microliter of the fluorescent substrate reagent. And now how the spin chip actually works, if you come closer you'll see that there are like holes which allows the liquid to be inserted to all the wells instead of doing them individually. As you can see here, just putting it through one of these holes, I have distributed the liquid in all of the wells. And now what I'm going to do is use 100 microliter pipette to use the individual bacterial samples into the wells separately. And each well consists of 40 microliters, or that is basically the capacity. And then basically use a blank as well. And after that, what I'm going to do is simply rotate the chip, which allows throughput to carry the sample or the fluorescent substrate reagent towards the samples and give like a mix. And what happens is I'm just going to leave this here for a couple of hours. I've noticed that like doing some shaking or incubation on shaker helps, but even without that, it worked pretty well otherwise. So we're just gonna leave it.
So yeah, that was just like a demonstration to show the mixing process of the chip. But to understand how the detection actually works, let's understand a little bit of the background biochemistry of it. And to understand that, we need to know what a Lac-Opron is. And the Lac-Opron is basically a group of genes which are required for the transport and metabolism of lactose in E. coli and other enteric bacteria. As you can see here is a structure of that. And the Lac-Opron consists of Lac-Z, which is a subsequent protein of the Lac-Opron, and Lac-Z's gene product is an enzyme called B-Galactosidase, or beta-gal. Beta-gal is very important because one, it's obviously plays a huge role as its function is to convert lactose into glucose and galactose. But we are also going to need beta-gal for our detection, and I'm going to show you how. So for our detection, we used Abcam's Lac-Z beta-gal intracellular detection kit. And the kit contains the fluorescent substrate reagent called FDG, as I've already mentioned. And what happens with FDG is it shows a positive result with FDG shows like a yellow glow under blue light if there's the presence of E. coli. So how the FDG actually works, you can see using these structures. As you can see over here, the lactose is converted to glucose and galactose using beta-galactosidase. That's basically the enzyme that breaks down lactose to glucose and galactose. Now, if you put FDG in the sample, what beta-gal will do is break down FDG to fluorescein and galactose. And fluorescein is the chemical that basically glows under blue light, showing positive result of presence of bacteria. The protocol is pretty simple of performing. We obviously have to do centrifugation as we are using LB broth, which we have to like discard and kind of like resuspend the cells back in sterile distilled water, and incubate, moderate shaking, and stuff like that. So for performing the intensity or detection limit test, we've used the JASCO 8500 spectrofluorimeter to run the experiments to determine the fluorescence intensities. So here, as you can see, it's one of the runs that we have done using excitations of 488 nanometers and emission wavelengths of 512 nanometers. And the reason for that particular use was this gave us like the best results, or the peaks were actually near 512. So here you can see the intensities, various different intensities at various different time points. Here is a typical set of data. Well, we performed like 66 runs over like 10 or 11 time points. And here is the data plotted into a graph. Here you can see with the presence of bacteria, the intensity increases over time for the two E. coli samples, but for a blank sample, which is just the FDG with no bacteria, the intensity remained the same, showing that there is no bacteria in the sample. So for the next experiment, we performed the test in the spin chip, and as you can see, is a bright glow for the Lac-Z gene activity because of the presence of beta-galactosidase, showing that there is E. coli in these two wells, and then no fluorescence in the wells that are blank. And then I've performed some other detection limit tests for the E. coli using various concentrations of FDG to see how much we can go doing concentration ratios of 1 to 10, 1 to 20, and 1 to 50. And here you can see some of the results from those experiments. Obviously, a higher concentration of FDG or higher concentration of E. coli resulted in higher intensities over time. Some other concentration varying detection limit tests. Here you can see from like the previous test of using the different ratios starting from 2 to 1, 1 to 1, up to 1 to 100. You can see basically the gradient of intensities The increase in the concentration of E. coli going towards the outer wells shows us that there are more E. coli or higher concentration of E. coli at the outer well compared to the inner wells. So this like difference in intensities can be captured, and then machine learning can be performed to kind of like evaluate the results and show how many number of bacteria or the concentration of bacteria comparing to a standard stock image. And then I've performed a validity test without centrifugation, but obviously that failed because LB broth glows by itself even without the presence of bacteria. Perform another detection limit test going up to 1 to 1,000 ratios, and then which basically proves that we can do bacterial detection using only few nanoliters of the FDG fluorescent substrate reagent. Here you can see obviously the 1 to 100 ratio proving a lot higher intensities than the 1 to 200, 1 to 1,000 ratios. Here you can see multiple assays being performed simultaneously with the spin chip. Here obviously is the detection limit test showing a gradient of concentration, and here's the failed non-centrifugation test that we have performed. And up next is I want to show you the new design of the spin chip, or the spin chip 2.0. This uses far less material as it's smaller in diameter and thickness, but the advantage is it has a larger number of wells, 120 wells on each side compared to 60 on the previous design. Here we are using CNC machine at the machine shop to make the chips with the use of a technician called Greg Arnold. And that's basically the design of the new chip. That's pretty much it. I want to thank some people for my research presentation. Obviously, Dr. Du, first of all, for advising and supporting this research. Jimmy or Jiacheng He for making the CAD model, chip design, the Dymix experiment, and basically collaborating with me throughout the whole summer to perform a lot of these experiments and research. I want to thank you, Chang, for assisting me with the Teflon coating, and then another student called Wimit Desanayaka for some Excel data entry task, and obviously being the videographer for the experiment video. And here are some references, and with that, that's pretty much it. I want to thank the RIT Research Symposium organizers for giving me this opportunity to present my research. And if you have any questions, please feel free to reach out to me at rk6119@rit.edu, or check out our website at www.3n@rit.com. Thank you very much. Have a good day.